Hello, everybody, and welcome to your lecture on brain, mind, and education. For some of you, possibly, I don't know how many, perhaps this is your first lecture on your course. Oh, okay, just checking. We have an awful lot of technology going on here, and I think the first thing I should do is to actually make sure that the speaker, I don't think this is on, is it? Okay, slight technological hitch. Let's turn that on. You should be able to hear me a little bit better when this comes on. 1, 2, 3. That's a bit better, isn't it? Right, excellent. There's also, I should tell you that it's being streamed live publicly, so anybody can tune in and see it. And in fact, I want to say a particular welcome to 4 students from the Dominican Republic who are still waiting to get here due to visa complications. And it's also then recorded automatically onto YouTube. And you should already have received an email that will tell you where to find it. But I need to tell you that it's being streamed live, so that if you shout out anything, I can't prevent that from being publicly broadcast to whoever. I also understand that the slides and the sound are being recorded as well, which is part of the new university policy. So it's been recorded twice at the moment. And where do we get hold of those?
You will get them through your platform, and you can edit them, and then they'll be made available in a couple of days. Okay,
I won't be editing. I don't think I'll just put it straight on, I think, if I can. Okay, so fantastic. Now, these first 2 lectures, you're going to be tested on, okay, because the assignment consists of 3 parts. The first part is a multiple choice, everyone started writing now, is a multiple choice test, and it counts to around about 25%. It's on your knowledge of neuroanatomy and function. And the deal is that I'm not allowed to ask you anything which is not on a slide in the first 2 lectures. Okay, so only the first 2 lectures do you have to swat, but of course all the lectures are interesting, so I expect you to spend a lot of time looking at those. But I did notice last year that I had about 500 hits for the first 2 lectures, and then the number of hits sort of went down. It was a bit sad, but there you go. So that's important. I mean, I'll also be giving you little quizzes along the way as well, to sort of, you know, next week we'll try a few quizzes to get you warmed up for that. The other thing is that I always like to challenge myself in these situations, you know, whenever it's a new academic year. So my new challenge to myself is that I'm going to try to ask you 3 questions. So my challenge is asking you questions about the teaching and learning strategies that I'm using in the lecture and what they might be, what they might have to do with the brain. Okay, so I'll be doing different things, and then I'll be, you know, asking you at the beginning of the lecture to look out for these and say, well, what on earth has that got to do with the brain? Now today, you're going to be playing Simon Says. Okay, yes. You're going to be feeling a plastic brain, which I think have been distributed with the aid of my helpers. I can see some plastic brains are hanging around. And what else are you going to be doing? Well, something mysterious anyway, and I'll be asking you about it at the end. See if you've got any ideas about it. So the reason I'm doing that is because I want you to start thinking, we're all in education here, yeah? We're all interested in education. I assume you are, because this is an education department. And so it's really important, I think, to try to make links between what we're learning academically and what it actually has to do in real practice. And I think more and more, what we're coming to understand about the brain is relevant to how we teach and how we learn. And there's no better way of thinking about that than actually experiencing it. Now, my friends in the Dominican Republic, for them this is 11:30 in the morning, which is an excellent time to have a lecture. Unfortunately for us, it's 4:30, which is a terrible time to have a lecture. So I must give you little breaks as well. And you know, try to remind me about that, because if we start going into function, we shouldn't go into function before we've had a break, okay? So with neuroanatomy, then we should have a little break. Okay, so we'll start off with neurons, and then as we start, we'll have a little break, and then we'll go into sort of bigger regions of the brain. I'm also passing around a register, because actually, despite the multiple ways in which you can look at this in a virtual way, you are required to physically attend. So please do mark your name on here. Oh, are there 2 registers? Now that could be, oh no, there's just an awful lot of people. Okay, blimey, I've never seen that before. Okay, so there is just 1 register. Please, just in the first column, can you put your initials as it comes round? There's 2 pieces of paper. Try to keep them together if you can. So the multiple choice test is the first part of the assignment. I then ask you to choose a topic that's of interest to you, and topics that are chosen every year are things such as dyscalculia, dyslexia, ADHD, and you can focus on the neurocognitive function of that issue and review it. It's a scientifically based assignment, so it's less about opinion, I'm afraid. It's more about reviewing the neuroscientific literature, but that's really what this whole unit is about. It's about trying to empower you so that you can go to the neuroscientific literature and ask questions of that literature that are of interest to you. So we will ask you to prepare a little poster that allows us to have a look at what your ideas are, and you can share that with other people and see what direction you're going off in. And then the final part of the assignment, the most important part, is a 2,000-word essay. 2,000 words. And you probably think that's far too few words to write an essay, but more and more we're being asked as scientists to express ourselves concisely. So, you know, when I'm trying to get a paper into Nature or some such thing, you know, we're asked, oh, can you put it in 1,000 words or 1,500 words? So I think that if a message is important and you understand it, you should be able to put it in a concise form. And actually part of the task in that assignment is to leave things out and prioritize and think what the most important things are that you want to say. By the way, please do pass the brain around. If you want to feel the shape of it, that's fine. That's a good thing to do. You're allowed to fiddle and play, but I'd rather you didn't drop it if that's okay. So 2 books which are recommended. Because you're in an education department studying neuroscience, it might be a good idea to think about the links between those 2, and you can buy my book. The best student, well, I'm bound to say that, aren't I? Yes. The best student's guide to cognitive neuroscience is called The Student's Guide to Cognitive Neuroscience, and it's just been revised this year. So there's a 2015 version now. I believe it may come with a CD-ROM and other exciting things. So that's thoroughly recommended. You should have found where this unit is now in Blackboard. You will find that on Blackboard there is also a link, I hope, that takes you to last year's lectures, which are all on YouTube. There are. Okay, I was thinking, did I do that? Yes, I did. Okay. So there's a link that takes you to a page on the MBESC website, which is our research network, where all of last year's lectures for this unit are in YouTube form. There's links to them, and also all the slides for those lectures. Now, I'm always changing my slides. I'm sorry, but even if you looked at them on Friday, I've made a few little changes last night. I was just revising what I had to say, and I could not resist it. It happens all the time. But generally speaking, it's very similar to the content that was there last year, okay? Right, so let's get cracking. Let's start off. We're going to be looking at neurons and other cells, neural networks, neural communication, the action potential. We'll have a little break, then we'll have a look at nervous systems, neuroanatomy, Broadman areas, and start a little bit on function and where you find parts of the brain that are associated with particular human functions, particular cognitive function. Okay, so this is a neuron. This is the basic building block of the brain. It's a very important type of cell. It's an information processing cell. So we have cells that do all sorts of things, but the neuron is dedicated to information processing. It doesn't just exist in the brain. Actually, you have them in your spinal cord as well. You have grey matter in your spinal cord. Grey matter is made of dense, compacted neurons. And there are some animals that have neurons but don't have a brain, like an octopus, for example, has a lot of neurons, but they're not centralized. So we talk about it not having a brain. But wherever you find neurons, they are processing information. And a An important part of the most important part is the nucleus. The nucleus that starts off creating an action potential, which is a signal carrying some information that passes down the axon. Now, this is a little bit like a wire, and in fact, an action potential is an electrical signal. Like with electrical wires, you can see the insulation that we have on these cables here. We have that insulation because we don't want to lose the signal. We don't want to be electrocuted either, but we want to make sure that the signal is safe. So quite often with a microphone, for example, there will be a steel sheath around the signal that prevents it from being interfered with. Now, if we don't have that steel sheath, then we get crosstalk between neurons. And so that's a very important thing that myelin does. And there are different types of cells that produce myelin around the axon and provide that insulation. The signal passes down there to the presynaptic terminals where they jump across a connection, but they jump using a chemical transition. Okay, it's no longer electrical, it becomes chemical at that point, and it makes contact with the dendrites of another neuron most often, and so passes on to yet another cell. Now, let's think a little bit more about this action potential and how it actually is created. First of all, we have to understand that there is already a voltage difference between the inside and the outside of a neuron. Why is that? Well, it's because the membrane, the thing that actually separates, going around it, that separates the inside from the outside, has certain types of properties, and one of those is having selective ion channels. Ions are positively or negatively charged particles that allow some neurons to pass, others with more difficulty, some none at all, won't allow them to pass at all. And what we find is that at rest we have a situation where potassium ions inside the cell can easily cross. Compared with chloride ions and sodium plus ions, they have more difficulty. But the fact that these are crossing easily, but not necessarily back in the other way, that means there's a flow outward of positive charge, leaving a slight negative residual inside the cell. We also have a pump, which is pumping out 3 positive sodium ions for every 2 potassium ions that are allowed in. So again, there's an imbalance in that pump. And all of that adds up to a negative potential difference between the inside and the outside of the cell. It's about 70 millivolts, it's been measured, and it's called the resting potential of the cell. So that's the normal situation for a neuron. However, things can change, and something has to create that change. And most often, it's a charge, although it can actually, sometimes this can be created by a peripheral neuron, such as a retinal neuron, that will respond to light. And that can be the thing right at the beginning of the chain. But most often, what causes one neuron to fire is actually an input coming in from another neuron. And that input arrives, and it causes that electricity crossing from across the synapse, causes the cell membrane to just momentarily depolarize. So instead of being negative, it starts coming up towards 0. And at a certain critical point, things happen in that neuron. And when it reaches a certain level of positivity, remember, see, it's gone past 0 now, it's now going positive, all of these things are happening. First thing that happens is the sodium channels start opening, which means that sodium is now flowing in, which is making it more positive. More sodium channels open, it's getting even more positive. And then the sodium channels close when it reaches the peak there, and the potassium channels open, and the potassium ions start coming out. So then it starts reverting back again to its negative state. See, it starts repolarizing, negative, negative, negative. And in fact, it drops below the resting potential and comes back up again. And what has happened is that it has fired an action potential. And that action potential can now travel down the axon in a similar way. Let's have a look here. I think it might be, oh, it's actually passed, oh goodness me, it's working. So you can see it's actually passing along. See, that's inside the axon. That positivity is now passing down to the presynaptic terminals, and they'll connect on to dendrites of another neuron. These are the dendrites here. And so what we have is a mechanism whereby electricity can be brought about inside the neuron and then pass down its axon to another neuron. Now that's very clever because it means we can now have electrical circuits made out of complex networks. So neurons can arrange themselves into networks, and a signal coming in here, let's just imagine, for example, these are peripheral visual neurons. Some light has fallen upon your eye, and it started off this chain reaction, and the action potential is going down onto the dendrites of another neuron, down to the axon, onto the presynaptic terminals, onto another neuron, and you can see that it's basically flowing through the neural network. Now, what is important here is that actually the way in which this electricity is passed across that point there, and we call that point the synapse, the connection, but the technical term is synapse. The way in which that signal is passed across will vary from one synapse to another. Some will pass all of it through, some will hold some of it back, not just pass a partial amount of it through, just a little bit of it, let's say. And so what comes out of these three neurons here is different to what went into the network here, depending upon the strength of these connections. Change the connections and you get a different mapping from the input here to the output there. And this is how we think we think. Okay, this is how we think. When you are thinking, you are processing information. It may be input from your memory, or it may be input from what you're seeing in front of you, and that becomes processed. It gets turned into something else. The most common thing that it gets turned into in the animal kingdom is what? What do you think sensory information usually ends up getting turned into when it goes through all your information processing? Yes. Movement, action. That is essentially how life has evolved. We are information processors turning input, which is sensory, into output, which is movement. That's a large part of what it's all about. Okay, so we say that these electrical signals travel in a complex, interconnected manner, and of course it is complicated because you can change these synapses. If you change these synapses, you'll get a different output here. And you've essentially transformed a complex input made up of whatever signals are going on to all these different neurons, and that might be different because you can have loads of neurons at the input, and it gets turned into another signal at the output, which could consist of lots of neurons as well. But the way in which that happens depends on all these connections here, what they're set to, if you like. Although we often call them weightings, because of course a lot of people have a lot of fun with this idea in computer science. So this is how we create artificial neural networks. And a lot of computers and a lot of actually stuff you probably use now, I mean, when you get told on Google that people who like that also like this, and you get these adverts coming up telling you, you know, maybe you'd like to buy another vacuum cleaner, and you think, well, how do they know I'm interested in vacuum cleaners? How intelligent is Google? This is scary. Well, that artificial intelligence is being created by computerized versions of these sorts of neural networks. Okay, so now I've talked about myelin. There's a few phrases I have to introduce along the way because you're going to come across them in technical journals. So when we talk about afferent axons, we're talking about it going to a place in the central nervous system. So if I said this is one of the amygdala's afferent axons, it means it's an axon going to the amygdala. If I said it was an efferent axon, then it would be one of the axons that's going away from the amygdala. Okay, so there's a lot of this terminology, and I have to try to introduce it. So myelin insulates, speeding up the signal in between the nodes of Ranvier where there is no myelin. Loss of myelin can produce muscle weaknesses and seizures, most famously known in multiple sclerosis, but also viral infections. Just some common viral infections can have that effect as well. Myelin is whitish, and when we look at brain pictures, we often talk about grey matter and white matter. And I said that grey matter was caused by this very compacted, dense collections of neurons, neuron cells, but it's the white myelin around axons that give white matter its color. And that's why we have gray matter and white matter in the brain. We often don't look too closely at the white matter in the brain when we're looking at brain images. We tend to be interested in the neuron cells that are firing rather than the axons. But a lot of that is because the wiring is so complicated. But we're beginning to understand more about that now, and we're taking more notice of white matter. Sometimes when the neurons are very compacted together, we call it a nucleus. So one of my favorite parts of the brain is the nucleus accumbens, which is a desire part of the brain. And nucleus just means very compacted, dense neurons. Neurons are not the only cells in the brain. There are some other important ones. Most famously glial cells, which tend to be the housekeepers. So some of these, for example, remove excess potassium ions, leaked neurotransmitters. We'll talk about neurotransmitters at some point. We also have different cells that make myelin. Oligodendrocytes make myelin. One of those wraps around several axons at once. So you can see that's an oligodendrocyte because it's wrapped around this axon, but it's also wrapped around the axon of another neuron. And Schwann cells, but they tend to keep to just one axon, and they're most commonly found in the peripheral nervous system. Peripheral nervous system meaning, for example, the axons that are passing down your body, because some of these axons are very long, because they have to actually cause your muscles to operate down at the end on your foot. Sorry, I was thinking too much about the brain. I couldn't remember what the name of that was. But, okay, so, and then we have microglia, which are very important for cleaning up the central nervous system debris. And we find, for example, after strokes, we get a massive increase in the amount of microglia. Okay, so I have to put up some of these horrible sorts of diagrams just to help you sort of try and think about where you are as I'm guiding you through the nervous system. And, I mentioned the peripheral nervous system. That tends to be, it's not the bit that we're most interested in education, I have to admit, but actually it is very important for a lot of our behavior. It tends to be divided into two parts, the autonomic and the somatic. The autonomic is all about controlling internal organs and glands, heart, sweating, breathing, those sorts of things. And we then further divide that into the sympathetic autonomic system, which is fight or flight response, where you're actually increasing the amount of energy that you're using, and the parasympathetic system, which is more about actually reducing the amount of energy that you're using and going to sleep and resting and digesting. And then there's a somatic system, and this is very much about this idea of input-output. So you feel your, I won't keep going about my foot in case I forget what it's called. You feel information coming in there. You can feel your touch. That's information coming in through your hand. And then if you move your hand, well, that's information going out to your hand. Okay, and somewhere in the middle, if you feel a cup of coffee, you think, well, that's a bit too warm, then you've had to do some processing there to turn the sensory information into a motor response. Sometimes that can happen so quickly you're not even aware of it. But it still requires the brain, not always, because you can have reflex reactions that don't require the brain actually, but it's still, usually there's some sort of processing in the middle that does require the brain. Okay, so when I say it doesn't require the brain, it still requires the spinal cord, so it still requires grey matter, I think, to produce that processing. But the bit that we're interested in is the central nervous system, and the spinal cord is part of that, but also the brain, which what we're trying to focus on. And here it is. Okay, here it is, the brain. Now, I've got to try and help you navigate around the brain. And so it's important that you start learning some of these words. You know, unfortunately, neuroscientists don't like saying back, front, underneath, just to the side, which is a bit of a shame, really. So probably make things a bit easier. They have to use these complicated words. Well, not complicated, but they're a bit Latin, aren't they? A bit Latin. They're not the normal words. So there's a slight health warning with this diagram, because this is true for the human brain. But some of these words, like dorsal, for example, actually mean something different in an animal brain, because some of these terms depend upon how the head is being held. Okay, so because a cow is going to be down like this, you'll find that these terms are slightly different. Now, you don't have to worry about cows, so forget about that. You have to worry about humans, so you can pay attention and just think of these as equivalent. Dorsal, superior, it's the same in the human brain. Ventral, inferior, it's the same. So that tends to be towards the top, that tends to be towards the bottom. And actually, those words are used, I'm using them here in a gross sense. So I could say, well, the superior regions of the brain are being the ones on the top. But actually, we could be just talking about a small part. So this is called the temporal lobe here. And I could say, well, it's the superior part, the region of the temporal lobe. And then I'm talking about the top of the temporal lobe. Or if I wanted to be clever, I might say the dorsal region of the temporal lobe, just to upset you. Okay, but it's still true. And these are sort of used interchangeably. And the bottom tends to get referred to as the ventral or inferior. The front of the brain, this is the front, by the way. You know it's the front of the brain because of this thing here. This is the cerebellum. Do pass the brains around. Be passing them around. I want you to feel the brains, okay? This is really important that you feel the brain. Don't just look at it. Take it out of the stand. Feel the brain. And you feel that lumpy thing at the back underneath the brain, which is like a little cauliflower here, and that's called the cerebellum. And that's the first thing I see when I want to know, am I looking at the back or the front of the brain? I guess the second thing is this temporal lobe here, and you can see the crack sort of starts here and going up. That's the next thing I look at. So this is the front of the brain, and front we usually refer to as rostral or anterior. And we also have this word for the back of the brain, caudal and posterior. It gets worse. You're also going to come across these terms. Ipsilateral means the same side. So if I say, ipsilateral visual cortex, it also activated ipsilateral visual cortex. It means the same side of the brain. Okay. If I said contralateral, it would mean on the opposite side. So your hand activates, so moving your hand activates contralateral motor cortex. It actually activates motor cortex on the opposite side of the brain. Yeah. It's the same with visual processing, actually occurs on contralateral visual cortex at the back of the brain. Bilateral means both sides. So if I said bilateral frontal cortex, I'm saying, well, both sides of the front of the brain. But bilateral frontal cortex sounds much better. Okay. So medial means towards the middle. Okay. Middle. So the medial regions of the brain are actually between the two lobes. But again, you can talk about medial within the region. So sometimes we talk about medial temporal regions. Lateral is away from middle. So if I was saying lateral frontal cortex, then I'd be saying not frontal cortex here, but lateral cortex away on either side, away from the middle. Rather than just say near and far, we say proximal and distal. That's a bit unnecessary, isn't it, really? I think what is a bit unnecessary, but that's what they say. These are important because these are quite often used in reference to brain images. So people will talk about, oh, it's a sagittal brain image, and they mean down the middle, like that. Down the middle. Or they'll say it's a coronal brain image, which means like that. Or they'll say horizontal or transverse, Again, we've got 2 words for it, and it's like that. Okay, so let's play a little bit of Simon Says then. What I want you to do here is, you know, Simon Says. Simon says, put your hands on your head. You have to put your hands on your head. This is, I'm sorry if there's any cultural problems here, but this is a game. This is a game we play in England and Britain. I don't know where else it's played. Simon says, put your hand on your head, and you have to put your hand on your head. You have to do whatever I do, basically. Simon says, put your hands rostral or anterior. Okay, Simon says rostral or anterior. Simon says posterior. Simon says posterior. Simon says superior. Oh, very good. Simon says caudal. Sorry. Simon says ventral. Simon says superior. Simon says anterior. Of course. You caught me out there, right? Simon says dorsal. Simon says posterior. Simon says ventral. Simon says superior. Okay, oh yeah, very good. Okay, very good. Very good. Well, we haven't finished. Hang on. Right. Whoops. Simon says, now you've got to indicate here where the brain is getting cut. Okay. So it's either going to be like, you know, like that, or like that, or like that. Imagine that there's slice going through your brain. Simon, oh, what have we got up here? Simon, okay. Simon says transverse. Oh, very good. Yeah, Simon says transverse. Okay. Simon says sagittal. Simon says coronal. Simon says transverse. Simon says horizontal. Oh, very good. Simon says sagittal. Simon says coronal, sagittal, transverse, sagittal, coronal. Great. Very good. Very good. Have we got any more? Oh, yes, we have. Okay. How do we've got to try to indicate. Simon says contralateral. Okay. Contralateral, opposite side. So if you're using that hand, it's going to be that. Okay. Simon says bilateral, both sides. That's it. Simon says proximal. Simon says distal. Proximal. Distal. I like that one. Very good. Oh, that's a bit more boring. Okay. Okay, so we've already dealt with that. You can put that away now. I should have something that just covers that up, really. We're now on the brain and the spinal cord. And look, we're going to leave the spinal cord behind because it's really interesting, but actually we're mostly interested in the brain. And look, you can see the brain actually divides into 3 areas: forebrain, midbrain, and hindbrain. Do you know how long the brain has been dividing into those areas? How long do you think, in evolutionary terms, how long has the brain been dividing into midbrain, forebrain, and hindbrain? Yes?
I think during the pregnancy period is divided.
Sorry, when?
It is divided during the pregnancy period.
Oh, that's very good. Sorry. Yes, it probably, but I meant in evolution. How many, over millions of years, when was the first animal, do you think, that actually had a tripartite brain made of midbrain, forebrain, and hindbrain? There is a reason why I'm asking this, because sometimes there's some misunderstanding about this. Okay, so people talk about the reptilian brain, don't they? Yes, well, forget about that. Because our brains have been dividing into these 3 regions for, I think, about 500 million years, I think, since, and in fact, you know, since before fishes. So it's a very long evolutionary chain, but this basic plan has stayed that long. However, it does vary in size. And you can see here, this is most of the forebrain in a shark, and this is most of the forebrain in a human. And hindbrain includes the cerebellum there. You can see the cerebellum in the shark is almost the same size as its forebrain. And so it does actually change quite a bit from one species to the next. And you might think, oh yeah, primates, eh? Humans, we have the biggest forebrain, yeah. And we've done very well, and that's what it's all about, because as humans, we're really intelligent, and therefore our forebrain has evolved much bigger. Well, actually, you know, the forebrain we have is exactly what you'd expect in terms of its size of a primate with a brain our size. Okay, there's been no great evolutionary shift between us and other primates in that sense. Our brain is just bigger. But the fact that we are primates is actually really important, because we are very efficient at packing in neurons. So be grateful you're a primate, and then be grateful that you're a big primate. Okay, so the brain is the forebrain, the midbrain, and the hindbrain. And this is what the lady here was saying, actually, well done, that, you know, we see these changes develop early on. You can see there 6 weeks after conception, we're already beginning to see this forebrain, hindbrain, and midbrain developing. And eventually you end up with, this is a sagittal slice. Now I can say that because you know what it is, sagittal slice through the brain. And this midbrain is actually very small, isn't it, in a human? But the hindbrain is a little bit bigger, and the forebrain is pretty well massive. But one thing I do want to emphasize, because this is a slice through the brain, okay, you've got two hemispheres in your brain. You've got a left hemisphere, you've got a right hemisphere. But it's joined together by a series of what we call commissures, and the biggest one is the corpus callosum. And that's an information superhighway. So let's get away from this idea that you're left brain or right brain, because even for the smallest day-to-day task, you're going to be using left brain and right brain together, and in a very sophisticated fashion. And what allows them to communicate with each other is this corpus callosum here. So here we are, the medulla, the pons, and the cerebellum. The medulla, very important for maintaining breathing, blood circulation. Pons, important for relaying movement, regulating breathing and taste. And the largest part of the hindbrain is the cerebellum. We used to think it was just about motor coordination, so we had a rather sort of diminutive view of it. We sort of thought, oh, humans, really wonderful, because our forebrains are so much bigger than our cerebellums. But actually we found out the cerebellum is really important for a lot of cognition. It's very important for timing, for example. And actually, when you look at the number of neurons in the cerebellum, again, it's exactly what you'd expect for a primate of our size, with a brain our size. Nevertheless, you know, it is much more. It is very important for motor coordination. The midbrain is important for a number of reasons, not least because there are some important parts of what we call the basal ganglia. And the basal ganglia are very important for making decisions about movement. And again, that's something which is part of the vertebrate evolutionary plan since the very first jawless fishes. So there's two important parts of the midbrain. This is a section that's gone through the midbrain. The tegmentum, nuclei connected to motor control, and the tectum, controlling and relaying sensory information. So wow, you know, information coming in, senses, information going out, motor control. And as soon as, you know, we had two eyes and we were navigating, and I'm talking about us, meaning fishes, through the ocean, we needed this sort of process. We had to have sensory information in, and we had to give out motor output. So basal ganglia, very important, and this is a very important part of it. But of course, we're most interested in the forebrain in many respects. As educators, there are some very higher order thinking processes that attract us to the forebrain. But there are parts of the forebrain which do not involve the cortex. So mostly when you think about the forebrain, you think about the wrinkly thing up here, this big wrinkly part. But actually there are some subcortical parts of the forebrain, and that includes the thalamus, which is a sensory relay to the cerebral cortex. All your senses go through the thalamus before they go to your cortex, except one, which is smell. Yeah. And if you've ever wondered what, actually an example of this is that my son opened, he's 9 years old, he opened a packet of baby spinach the day before yesterday, and it was off, and it'd been off for some time, I think, and it was so difficult. Actually, I'm sorry, I should be thinking about it now. It's making me feel sick thinking about it now. It's coming back. But what I wanted to say was that it's very difficult sometimes to control smell information when it's entering the brain, because it goes straight into the brain. You know, we've evolved from olfactory mammals, mammals which are probably, you know, closer to rats, and rats do a lot of their processing around smell. That's the big thing for them. And so that's probably why our olfactory processing goes actually straight in to the front of the brain here still. Of course, our interest in smell has diminished. We're more interested in many respects in visual and hearing processes now. But, you know, go back a few 100 million years and it was all about smell. Oh, sorry. And then there's the hypothalamus, which is below the thalamus. That's what it means in Latin, below the thalamus. And that's about controlling our bodily state in respect of, you know, if you're hot, then you need to start sweating. So, you know, sensory information in, I'm feeling hot. Output, change sweat glands, you know, and start producing sweat so you can cool off. And then, of course, the bit that we all see when we all think about, and the bit that's most obvious on the human brain is the cortex. And here's the wonderful cortex, but but don't forget these very important subcortical structures. For example, the striatum. This is the striatum here, and made up of the putamen and the caudate, and that's very important for our motivations. And we'll talk more about that in the emotion lecture. My friend, the nucleus accumbens here, the more that activates, the more you want something. Sex, drugs, and rock and roll center in some respects. The amygdala, which is very important for emotional processing, and it's hanging on the end here of the hippocampus. And the hippocampus is very important for memory formation. So you can see what's going on there. The brain has evolved things together that need to work together. And our emotions and our memory quite often need to work together very quickly. So that may be why those are together. What else did I want to mention? That's about it. Yeah, just to mention that, you know, the interest in the subcortex is so intense that I went to a meeting the other day on reinforcement work, learning, and one of the experts there was saying, you know, the cortex, it's just a duvet for the rest of the brain, basically. So it's a bit more than that, but you know, but it just shows the way in which scientists' views have changed. Actually, the cortex is very much a general processing, domain general piece of kit. You know, we do find that parts of it become specialized in particular function, but if you lose that part of the cortex that was specializing in it, another part of the cortex will probably pick it up. Okay, so where are we now? We got to the forebrain. Good, we got to the forebrain. Cerebral hemispheres. So we have two cerebral hemispheres separated by the longitudinal fissure. You can actually see through it there. It's interesting. And of course, the reason why you can't see the below it. Oh, I can just do that actually. I just realized, like, that's really satisfying. You press the button in just the right place. Oh no, it doesn't. Okay. The reason you can't see down the bottom is because the corpus callosum is there connecting the two things together. So if he's going to stop, that's so disappointing. I thought I was doing that. Oh, never mind. Hey, you see the corpus callosum is just there. So these wrinkles are important because they're a way of finding our way around the brain as well. And we have a special name for them. We could have called it a ridge, but we don't. We call it a gyrus. Okay. We could have called it a valley, but you know, we don't. We call it a sulcus. So the ridge is the gyrus, and the plural of that is gyri. The valley is called the sulcus, and the plural of that is sulci or sulci, depending on whether you say tomatoes or tomatoes. It's highly folded. That's also worth remembering. The original area is double the page of a tabloid newspaper. So, you know, again, that's not something that's happened that's particularly human. It's the way in which the mammalian cortex is basically, that the more it grows, the bigger brain you get, the more folding you're going to get. Now, 4 lobes, and I think it's important, you really should be passing it around now because I want you to feel the frontal lobe. I want you to feel the frontal lobe here. This, as you pass it around, you can split it into 2. That's a better way of doing it. Maybe you get it further. I hope I don't end up with 9.5 brains at the end, but do split it into. The parietal lobe is just behind the frontal lobe. So frontal, you know, if people, I've heard neuroscientists ask, what's the most important part of the brain for education? And, you know, they usually say, oh, frontal. So traditionally, that's the bit that we're most interested in, the frontal lobes. Actually, I really want to challenge that. I think that's a bad way to go. The frontal cortex would be useless if we didn't have the subcortical structures underneath it. The temporal lobe here, auditory processing, but also its medial side, very important for memory and language as well, the left temporal lobe. The parietal lobe here is very important for bringing together different types of information and processing them. And the occipital lobe, strangely right at the back, is the part of the brain most associated with vision. And again, you have this contralateral issue whereby if you're seeing something on the left, it's the right part of the occipital cortex that tends to get involved most. And then, of course, there's this hidden cortex. Now, as you open up the brain, if you look inside the brain, I've got one here. Mine's more fun because it comes into more parts, but actually it creates more problems for me because I sometimes have trouble getting it back together again, I have to admit. Stay. So inside the brain, you have a lot of cortex, which you can't see from the outside. In fact, there's actually another island in here, which is called the insula. If you look inside there, you can see there's another little crack in there with some cortex hidden inside there. It's called the insula. But, oh yeah, I can't get it back now. But this is probably the most famous piece of hidden cortex. Insula means island in Latin. It's the cingulate cortex, which goes across here. And one very important part of it is the front, the anterior cingulate, they call it. And that's very important for control, attentional control. So you'll come across that quite a bit. Whenever anything is difficult, novel, right at the moment, because you're concentrating on me, your anterior cingulate is going, ooh, the blood flowing up there, trying to keep up. Well done, keep it going. Okay, so what I'd like you to do now is a little test for you. I hope the pictures I've given you allow you to do this. It's also a bit of I think it's probably a good idea to have a bit of a toilet break, actually, as well. Yeah, I'm going to have a little toilet break here as well. So have 10-15 minutes. Let's, you know, we'll come back here. I haven't got the time. What's the time? 20 past 5? 25 past 5? Somewhere around there, is it? Is it? I don't know. So let's say 22. That gives you 15 minutes. Go and have the toilet, go to the toilet, come back, have a scribble, see if you can fill these in your colouring book. You should have a little piece of paper there, that's your colouring book. See if you can identify where these things are. You've got the tools now to be able to do this. You know what inferior, frontal, sulcus, you know what that means, don't you? Okay? Oh yes, please do. Where's the register gone? Anyone seen the register? Oh, horrible thing has disappeared. Anyone seen the register? Anyone seen the register? Anyone seen the register? Anyone seen the register?
Yeah, I received that. Yeah, I did.
Oh, okay, yes, you're very welcome.
You provided a learning opportunity.
Oh no, you're very welcome to be here. Very welcome.
Extra sheets with that picture.
You know, I had 100 photocopied. It's on Blackboard though, so you can. Oh, sorry. It's on Blackboard though, so you can download it. Okay. I'm sorry about that. Anyone seen the register? Let's put it back, have they? Anyone seen the register? Register, anyone? Anyone seen the register?
Oh yeah, I see it, but
Aha! But I said, oh, my brain. Okay, no, that's good. Thank you very much. No, that's good.
Everyone that way found it. People that way found it. People that way.
Sorry, there are bound to be some people who haven't, so I'm just going to wave it and then put it up. Anyone not signed the register?
Yes, please. Thank you.
Scary, isn't it?
The dorsolateral prefrontal cortex.
That's not a bad guess. That's probably pretty much where I put it. Dorsolateral prefrontal is a bit. It's more like a region, you know. It's pretty. Basically, it's yeah.
It's here?
Yeah, basically, it's there.
So like,
You probably have to sort of, you'd have to go.
okay.
It's not one.
Sure. Let me ask you something. You said that the temporal is about memory and language.
Yes.
Here is vision, and what is hearing?
Yeah, but well, the thing is, there's so many. I mean, frontal cortex is involved with any higher level reasoning and decision making. But parietal cortex very involved with bringing together information from different parts of the brain. So multimodal association. So bringing together touch, vision, hearing, all comes together there. But also mathematics and also attention. There's yeah, I mean, functions are so distributed that it can be misleading to say that it's just about that. So you have to be very careful, really. Get a glass of water, I think. I'm just getting a glass of water. Right. Okay, so let's return. I've pulled one of my wires off. I think it's all still working. I hope. So let's just see if we got some of these correct. They're not completely straightforward, so don't worry if you didn't. Inferior frontal sulcus. Inferior means at the bottom, but frontal means frontal lobe, and sulcus means valley. So it's the lowest valley traveling along the front. Okay, so it's not always obvious because some of the cartoon drawings that you have to mark these on don't. But they, generally speaking, you know, these things exist, but they're often not. No brain is identical. So you know, they exist in slightly different positions in different brains, but you should find the lowest valley traveling along the front is your inferior frontal sulcus. The superior temporal gyrus, well, temporal means temporal lobe, so you should be looking in the temporal lobe. This is the temporal lobe, and the highest ridge traveling along the temporal lobe. Well, that's the highest ridge there, so it'll be somewhere along there. Okay, that's the superior temporal gyrus. What about this one's a region, the dorsolateral prefrontal cortex. Right, well, lateral means both sides, okay, or away from the center. But in this sense, it's a bit more bilateral because I think it really means really more both sides. But dorso means the top. Frontal is a frontal lobe, but prefrontal is just behind the front. It's before you get to the absolute tip. So dorsolateral prefrontal cortex, it's a very famous part of the brain. So you're going to come across this abbreviation a little bit, DLPFC. It's worth remembering. It's very important because it's involved a lot with working memory. So when you're trying to hold telephone numbers in your head, or you're having some strain on the amount of information you have to hold in consciousness, your dorsolateral prefrontal cortex may be activating more than usual. The superior frontal gyrus, superior means the top, frontal means frontal lobe, gyrus means the ridge. So you're looking at a ridge which is as close to the top as you can get of the frontal lobe. So it'll be basically this ridge here, right? Not in the middle, because then you start talking about medial prefrontal cortex, but actually here on top. The anterior cingulate we've already talked about, because anterior means front, cingulate is the island that's formed in between the two hemispheres. So it's going to be here, the anterior cingulate. Very important part for attention. Did you get any of them? Okay, well, don't be worried if you struggled a bit. You'll get more used to it. But it's useful because these names will be in the literature, and that's why I ask you to do this. There is another way of talking about regions of the brain, and you will come across this as well. Broadman classified all the different regions of the brain in terms of their fine cellular architecture. So you could see under a microscope perhaps, or maybe even without the microscope in some cases, that the texture of the brain is slightly different here to how it is there. So he was able to delineate all these different regions that shared the same granularity, the same texture of cell structure. And as a result of that, we now have lots of numbers, Broadman areas, BA8, BA9, this is how we refer to them. And we find that they are associated with particular types of function. Remember that all of these types of function are very primary. They're not functions like, you know, opening a door. They're not functions like going home for tea. They're very primary functions like, you know, primary somatosensory cortex, which means if it's primary, it's one of the first regions that actually processes that information coming from touch. Okay, and we find that Broadman's area 1, 2, and 3 are particularly important for that. Other regions will be involved, but BA 1, 2, and 3 particularly involved. Other famous BA regions, well, of course, primary visual cortex, BA 17, BA 41. You don't have to remember these for the test, okay? Really, you won't have to remember any Broadman areas, but I do want you to know that they exist because you'll come across them, and you'll probably find yourself going back to this slide or to Wikipedia, where I managed to get this diagram from, to find out where these Broadman areas are. The reason, by the way, I use so many images from Wikipedia, well, there's two reasons. It was just because I wanted to make sure that I could make these slides public so anybody could look at them. But then I found that actually Wikipedia's got much better diagrams than anywhere I could ever find anyway. It's really, really wonderful in that sense. So let's talk about function. But let's please be very careful. We do not want to go back to the Victorian times here, where they separated the brain into different regions, and actually by feeling the bumps on your head, they thought that they could tell you something about issues such as parental love, wonder, hope, wit. Where's the wit section there? Number 20, that is. Let's find wit. Oh, it's there. Okay. So this is your comedy area. Doesn't work like that. Okay, we know now that these are very complex things, and actually those functions are distributed across the brain. You employ very many different regions of the brain. So we must be very careful when we talk about function. We say that these regions are associated strongly with this type of process or function, but you know, over the thousands of years, we've decided to talk about human behavior in particular ways. And we think that comedy or humor is a really important feature, but that doesn't mean that it's represented in a little box. Like creativity is not represented in a little box in the brain. Okay, we've found it convenient to talk about it like that, and psychologists have found it easy to talk about it, some things, by making up box and arrow pictures, like working memory. You're going to come across this issue. Working memory, Baddeley and Hitch model, really nice, not in the brain. Okay, it's not there. In fact, you know, we find that working memory is also distributed in the brain in many different regions. So, you know, it's important to talk about function because that's what we're interested in. But let's not start thinking that we have little boxes that do different things. Okay, so just to illustrate this, just think about picking up your cup of soup. I want you to visualize this. It's very important that you visualize this. I'm going to ask you why you're visualizing it at the end, my teaching and learning strategies. I want you to visualize, you're reaching out, you're going for that cup of soup, okay? Think about doing that for a minute. You're picking it up. What's going on in your brain? Probably not very much at all. You don't think there's very much going on in your brain. But actually, if you could see the brain image of your activity in real time as it's really occurring, it would be like cascading, shimmering light across your brain incredibly fast. All many different regions of the brain illuminating, passing activity from one region to the next. I'm going to give a very crude description of that now, just to illustrate how complex this is, but it's probably much more complex than this. Okay, so motor and somatosensory, that's touch and that's movement. They are topographically organized. What does that mean? It means that the part of the cortex that deals with touch from that finger is next to the part of the cortex that deals with touch for that finger. Same with the motor. The part of the cortex involved when you do that is next to the part of the cortex that activates when you do that. So they're actually, the organization of them represents the way in which they're organized on your body. It's good also to have the touch and the motor together, because you have to operate those two very closely together when you're picking up your soup. You have to be feeling where your fingers are around the cup and you're lifting it up. Yeah? Do it too tightly, cup of soup. You have to coordinate your touch and your movement. Information from the thalamus, yes, because we haven't even got to the brain yet. The information is just coming through the thalamus at this point, except for the smell, of course. Oh, that smells good. It's going straight into the cortex. But you know, the vision, okay, and the feel, that's actually going to the thalamus. And maybe you can hear your soup? You can hear the croutons crackling, maybe. And there's going into a unimodal somatosensory association area. So, you know, straight away, it's going into the somatosensory area here. And that's beginning to send you some information about the, I'm just looking at this because it looks slightly different to the slide that I had before. I think I must have done this better somewhere else, and I haven't. Never mind. So the touch is coming in here. The movement is coming out of there. The touch here is being. Oh no, I see. It's going to this part here, where it's being put together in the parietal cortex, because you've got 5 fingers and a thumb wrapped around the cup. Okay, and that information in each individual packet of information is pretty useless in terms of understanding the shape, but you have to put those packets of information together to work out what the shape of the cup is. Information from that finger, that finger, that finger, that finger, and your thumb to work out what the shape of this cup is and whether in fact you're gripping appropriately. So somewhere the information from all that touch has to come together. So it comes together just behind the somatosensory area here in a unimodal sense. Unimodal means one mode because we're still just in touch here. But there'll also be visual information that's coming into the visual cortex. So let's not forget that as well. Because sooner or later, you know, it can feed to premotor areas, preparing movement in respect of feeling the shape. That's important. And then from there to higher order areas for more posterior unimodal associations areas. So posterior means back, yeah? So I'm going back in the parietal cortex here to more unimodal association areas, but it also has to make contact here in posterior parietal, where there's information coming in from temporal association areas in terms of timing, but also visual information is going to be coming back here as well into this multimodal association region. So here, there's information coming from about timing, about touch, about memory as well, what happened the last time you picked up a cup of soup, and touch is all coming together in a multimodal association area here in order to put together a single concept, the concept of cup of soup. Because cup of soup is not just the shape. Cup of soup is the shape, the smell, the vision as well. All of that has to come together for you to understand that this is a cup of soup. And of course, parahippocampal regions here. I remember the hippocampus, very important for memory as well. So that's bringing forward what happened in the past with your cup of soup. All of that has to come together for you to think about what you're going to do next. And all you've done so far is just reach out for your cup of soup. You haven't even, you know, lifted it up yet. Your motor cortex has just been preparing to do that. So premotor cortex will be activating at that point, Broadman area 6, just beginning. And already all those parts of the brain have activated, and probably many more. Actually, I haven't put in the visual cortex. I don't know why. I must have done this another diagram somewhere that's better. But, you know, it's complex. So simple things activate vast ranges of the brain. That's the important thing to remember. How do we locate function in the brain? We have several different methods. One that was very popular, becoming a little bit less popular, is positron emission tomography. In this technique, you ingest in some way, that's like eating or whatever, radioactivity, and that radioactivity is connected to molecules that the brain likes, and it may be a particular part of the brain that likes them. So it could be attached to glucose, and that's going to be used up as energy by neurons. But you can also use particular labels for neurotransmitters. So you could connect your radioactivity to dopamine, for example, and it would then become more involved in dopamine tracts, regions of the brain where dopamine is being used. And so you'll be able to pick up images from the radioactivity that is emitted from where that radioactivity is collecting. Now, because it's radioactivity, there is a safety issue with this, and there is a limited number of PET scans that you can have in your lifetime. So that makes it difficult from a research point of view, because actually we don't really want to be using up the number of times you can have a PET scan doing research, just in case you have an accident later on. So it's not that popular. The other reason why it's becoming less popular is because of the resolution. You can see that the resolution is not that great compared with an fMRI image. But it is useful because you can label particular messages in the brain with it. And so it still has a usefulness in that respect. So you can accurately target particular regions of the brain using chemical messengers and attaching the radioactivity to those. But it's got poor spatial resolution, which is why it's all fuzzy. And actually the temporal resolution in terms of the time is not that great either. You're talking about minutes. So you can't see the way in which the brain activity changes from 1 second to the next. It's just not possible. Something that is very good at temporal resolution is electroencephalography. And this works by picking up the electrical activity, the changes in the electric field caused by those action potentials moving down the axons. Now there, what you've essentially got is a charge moving in space. And if you've got a charge moving in space, then you're generating an electrical field that in fact it's possible to pick up. So if you put lots of electrodes here, you can detect changes in the electrical field that are directly related to how your neurons are firing. And that is instantaneous. So it's got fantastic temporal resolution. You can see things happening in the order of tens or 20s of milliseconds. But the spatial resolution, not so good. Because for one thing, it tends to be better at picking up the cortex, but what's underneath the duvet in terms of the amygdala and the reward system and those areas, basal ganglia, not very good at picking those up. The thalamus, not very good. So it's not bad for the cortex, and it gets worse for subcortical structures. And what we find when we analyze the changes in the electrical field is that there are particular wavelengths that change. And some bandwidths, the lower ones, for example, or sort of the alpha band between 7 Hertz and 14 Hertz, we find that appears to be stronger the more relaxed you are. Other ones, like beta, higher frequencies between 15 Hertz and 30 Hertz, they tend to be more dominant when you're particularly worried or stressed about something. And then there's the theta ones, which is the lowest frequency ones, which appear to be stronger when you're relaxed and you're meditative and you're in a creative state. So quite often for creativity research, they encourage people to change their alpha theta ratio. So in fact, it's not like you're going to sleep, but you are becoming more meditative and more creative. And people who focus on their alpha theta ratio and look at a screen, because you can see this in real time, they've found that music students at the Royal College of Music in London can increase their performance grade in their final exams by a whole point. So instead of getting a 2-2, they get a 2-1. Instead of getting a third, they get a second in their final degree examinations if they've been using alpha theta feedback. So this is very interesting. Okay, so this is my preferred tactic, is functional magnetic resonance imaging. This works by essentially looking at changes in the magnetic field due to uptake of oxygen in the blood. So when oxygen is taken up in the blood, electrons change their spins, and that means they're responding differently to a magnetic field. And you can see those changes in the magnetic field by pulsing the magnetic field at a radio frequency and seeing it relax back. And by the way in which it relaxes back and the way in which the magnetic field responds when you pulse it, you can tell that something is different in the magnetic field. And so you put a brain in there, and by looking at, by analyzing the way in which the magnetic field behaves after you've pulsed it, you can tell the anatomy, and you can also tell how much blood oxygenation there's been, how much function. So when a part of the brain becomes active, it uptakes more oxygen. We call that the bold signal, blood oxygenation level dependent signal. And depending on how you arrange the settings of the magnetic resonance scanner, you can look at anatomy or you can look at function. Or usually we look at both and put the function on top of the anatomy to make a pretty picture. That's how it works. Its resolution is quite good, so you can see detail in the brain. It can be a little bit uncomfortable because you have to go inside this tube. You have you have a sort of a secondary coil put around your head here. That's the secondary coil. But I don't know. It's not that bad. Has anyone in here had one of my scans? Yeah, they are. Liz has had one of mine. Yes, that's right. So I'm always looking for volunteers to do it. Was it that unpleasant? You wanted to go to sleep. Well, going to sleep is a problem, actually. So we have to ask them to do things to make sure they're awake sometimes, even if they're resting, you know. So, yeah, functional magnetic resonance imaging, very good spatial. Temporal is not so good, but actually we are developing techniques to look at that more closely now. And in fact, we can do event-related experiments where we see the changes when a particular event happens in a trial. And we've just got it opened in 2011, I suppose that's 4 years ago. We've got a brand new scanner now at the University of Bristol at the Clinical Research and Imaging Centre, and that's where I do some of my research now. Okay, so in summary, we've looked at neurons and other cells. We've looked at how neurons add together and make neural networks for information processing. We've looked at how communication occurs within those. And that's the basis of thought. That's how we think, okay? That's what we're assuming thinking is all about. It's about information processing in the brain, and it happens through these networks of neurons. We've looked at key orientation terms to find our way around the brain. 3 parts of the brain: midbrain, hindbrain, forebrain. We've looked at the cortex, the 4 lobes. And we've looked at some subcortical structures as well. We've talked about the amygdala and the hippocampus. There is this other way of looking at regions, which is Broadman areas, which you will come across. And we've looked at some very, very simple associated functions, really just to emphasize to you that even the simplest tasks activate such a broad distributed range of regions. And we've looked at some of the methods we use to identify which parts of the brain are activating more strongly than others. Now, what's neuroscience got to do with education? Well, why did I pass around a plastic brain? I don't know if you've given this any thought, but it's actually because it helps you understand the shape. That's why I wanted you to feel the brain. Okay, feel the brain, because shape is very important in terms of finding your way around the brain. There's 2 ways in which you can understand shape. One is through vision, but the other one is through touch. And those 2 sources of information come together very early in the brain. And in fact, these green areas represent parts of the brain which are activated both by touch and by visual processing. So it's almost as if vision and touch are sending the same sorts of information. Well, at some point in the brain, it does become the same sort of information very early on in the brain. So by giving you a visual stimulus, I'm actually being able to see it, but also touching it, it helps me communicate much more efficiently, much more easily about the shape of the brain. And you need to understand the shape if you're going to find your way around it. However, beware, I don't know if we'll have time to talk about this, but vision and sound are not like that. Okay, vision and sound come together in a much more complex way in the brain. And you probably, when you think about it, that probably makes sense because, I mean, for example, when I hear a loud exhaust, what do you think about when you hear a loud exhaust? What is the vision that comes into your mind? If you hear a car going past, what do you think about? Without turning around and looking, what are you expecting to see? Because you can't see the exhaust actually operating in the car, so therefore your brain has learnt a whole load of complex ways in which you're going to relate that sound to the vision. I think about teenagers. I think about broken exhausts. I think about MOT bills. those sorts of things, having to go to the garage. Now those, you know, are going to be different for different people, and that's very important, okay? Because if I was a teenager, I'd be thinking, yeah, loud exhaust, you know. Maybe that's important when you're 18. When you're my age, maybe it's important to be thinking, oh, another bill, you know. So we have to learn more complex relationships between sound and vision, but that can also be very helpful in an educational sense. But when it comes to shape and vision, they come together really nicely, really early on. And so it's another way for me to communicate multimodally the idea of shape. Why did we play Simon Says? Have you heard about the enactment effect? That when you do something, you're much more likely to remember and understand it. And of course, in a way that's obvious because we have evolved with brains that essentially process information in, sensory information, and turn it into movement, turn it into action. That's how our brains have evolved over millions and millions of years. And yet we have an education system, of course, where we expect quite often people just to sit still and listen. So it's very important if we can involve action in it. Because the enactment effect can be measurable, and we're beginning through neuroscience to understand it a little bit more. That's a lamprey fish, by the way, which is one of the earliest, well, it's related to what we think was the earliest ever vertebrate that had one of a brain not dissimilar to our own, very much dedicated to this idea of senses being turned into movement. Enacting helps you remember. Why did I ask you to imagine a cup of soup? How did that help you learn? Because when you imagine, if you strongly visualize, actually, even if you don't strongly, if you just visualize, you use almost as much of your visual cortex as if you were actually looking at it. So imagination is a really important part of learning. So visualization and imagination is potentially a very important learning tool. And we're beginning to understand that a lot more from neuroscience. Because when we can see people's brains activating when they're imagining. And the way in which the brain activates is incredibly close to if they were actually looking at the thing. So you don't have to take them to the Himalayas, you just have to help them imagine what that is like. And they are in the Himalayas. So that's another really important thing that I wanted to sort of communicate to you. Right, we have a little time for questions. I like to leave a little time for questions because there are many things that I brought up today which you might want to ask me about. Yes.
Is it the EEG? Have you ever heard about the neurofeedback?
Yes, so neurofeedback is quite often, I'm being asked if I ever heard of neurofeedback. So actually that's what the music students were using. They were using EEG, and they were looking at their alpha and theta, which is being transformed online into an image on the screen that they could use to change their mental state. And they were changing it more towards a point whereby they could think more creatively and respond more sensitively. And it seems to work.
Yeah, I'm having trouble to understand. They use it for autism lately.
That I don't know about the success of it with autism. I couldn't tell you about that. I think most neurofeedback is EEG based. Yeah.
And the last question is, do you have the reference for
the person to look at for knowing more about that is Griselier. It's a G-U-R-U-Z. Sorry, something's gone wrong with the camera. G-R-U-Z-E-L-I-E-R. That's John. He's at Imperial, I think. Yes. Is that a hand? No, it's not a hand. Yes.
It might be that question. Why is it that we've got such a large part of the brain where vision is
Right. Why? Well, I mean, I think one of the issues is probably, I mean, what changed, I think, really was a huge meteorite hitting the Earth. And that's really when mammals started developing much better visual processing. So it's after that break in the evolutionary record, really, that we start beginning to see a development of other senses other than just smell. But we certainly started off as olfactory mammals, and then these visual processing started becoming a lot more important, really, as we start possibly. I'm not. I mean, we don't know, do we? But as we develop more sophisticated hunting and ways of capturing food, I should imagine. Yes.
Can you please clarify the difference between MRI and fMRI?
Okay, that's a good question. So we talk about MRI, magnetic resonance imaging, and that tends to be the so when we say MR, that's magnetic resonance, and sometimes people say magnetic resonance imaging, and then sometimes people do this. They put a little F there, and that little F just stands for functional. So in fact, if you're doing anatomy, you can talk about anatomical MRI scanning, and you just set the scanner to pulse in a different way so that you can look at the way in which the magnetic field is responding in a way that's more sensitive to anatomy. Whereas you set it in a different way if you're doing fMRI, if you're interested in the blood oxygenation. So that's the only difference, is the F, which stands for function. Yes. No, I love stupid questions because they're never stupid. No pressure.
What determines the peak on the action potential? Why does it change?
Action potential. Why does it change?
Yeah, and the neuron.
Well, I mean, the cell is designed to change its potential, and at the same time start producing processes. It starts processes to reverse it, because obviously, because what you have. No, it's a good question actually, because maybe I did not make this clear, and I don't think I did. Is that you get a pulse? So we talk about the message. The message itself, the electrical message, is actually one firing of the neuron. So it goes bang like that, and it's just one like that, which then goes down the axon. But you don't want it to go up forever and ever. So as soon as it gets to a certain point, it has to start changing. The potassium channels open, and then that starts reversing the process whereby it's depolarizing, and it goes back down again. The design of the cell. Yeah, no, the design of the cell, which, so, we've had, if you go back, I'm quite interested in evolution because I'm supposed to be writing a book about it at the moment, but if you go back to, 3.5 billion years ago when life began with bacteria, in fact, even with bacteria, you see them responding. So E. coli, for example, they actually show a behavior which is quite intelligent. They can find their way towards food in your stomach and away from poisons that might kill them. And they do that using ion channels, and it's those ion channels that actually, similar types of ion channels in the neuron, that do this rather complicated thing of responding to a positive pulse coming in by sending a positive pulse out. Yeah, I'm not sure who's first, let's say, yeah.
Thank you. You were talking about the EEG, right? And you said something about hertz, and if I remember the physics.
Oh, yes, good point.
You're basically measuring oscillations.
Yes, it's cycles per second.
What cycles are we measuring here?
The actual cycles. So if you take if you look at the EEG signal, it's just noise going like that. But then if you filter it, you'll find that noise, because all noise is actually made up of frequencies that are all added together. So you just put it through a filter, a bandwidth filter, and then you can see how much energy is in that restricted bandwidth in that noise. And within a certain frequency range, we call that alpha, because it seems to be related to certain types of behaviors and brain response. Yes?
Oh, this is again on the subject of action potentials. I saw an animation on the internet. This is not a good start. Basically, it wasn't just the one pulse. It appeared to be many pulses going all the way down the axis.
Yes, so that's because it keeps firing. So if a neuron is getting very excited, it's going to be basically the excitement is measured in the number of times it's firing. So it's not the height of the action potential, that's always the same. But the information that differs is actually how many of those pulses are actually going down. So it's a good question to ask. I don't think I made that clear. So if we say, oh, this neuron is getting very excited, it's actually producing a lot of action potentials. It's not producing action potentials of a different size. Yes?
So one of the neural networks, and that's how they think we think. Do you mean that in terms of just processing of information, or actually in terms of interpretation? And
Okay, so what we've done so far is talk about how we think, how the brain thinks. But what we haven't done is talk about how the brain learns, because when we learn, we change how we're thinking. So I've said that the brain can do some quite clever things because we have these neural networks whereby information coming in gets transformed into a different information coming out. So it could be that when you get light coming into this eye, it's really important for you to be moving your right hand, let's say, or your left hand. And that can happen because those first neurons in that network are peripheral neurons that take in the light. They send those pulses, those action potentials, through the neural network to the other end, which is connected to muscle, and it moves the right hand, okay? But that is going to be the same every single time you get light in your left eye. And of course, we have to learn different behaviors. And to learn different behaviors, that means we're going to have to find a way, which we'll talk about next week, of changing the information flow. And you do that and the brain does that by changing the synapses, okay? So that it might reduce the efficiency of the synapse. So in fact, you have to have 100 pulses coming in for it to shoot 1 or 2 out. Be one way of doing it. Or it might increase the efficiency of the synapse. And then that changes the information that you get at the other end. So you've got your input coming in, you get your information processing, you get your information coming out. And that's very clever, but it's not learning. To learn, we have to find some way of changing those weights and changing what comes out of the other end. And that changing is plasticity, that's neuroplasticity. And it happens, the bit that we're most interested in is the synapse, because that's where most of that neuroplasticity happens. Although another important part of it is actually growing new ones, new synapses. And we're just finding out that you can grow new neurons as well. But that's still an area we're trying to know more about. Any other questions? I'm quite happy to answer these because I think that everybody's thinking. I've realized now that there's a couple of things I didn't say. Yes?
When single cell recordings, do they actually just monitor one neuron?
Yes.
Just one?
Yes.
How big is a neuron? How do they manage to just get into it?
I don't know, but you see them on these microscopes with a little needle going in. I think it's absolutely amazing. Well, that's just incredible. Yeah.
What you said about visually working together when
Yeah.
when the information I've heard lots of mixed ideas about different learning styles. Is there any evidence that actually supports that some people learn better in a visual way?
Well, thank you for asking that question. So I can because actually we've just had a workshop today with the neuroscience and education people to talk about learning styles. So we do have preferences. If I give you out a questionnaire now, some of you will say, oh, I like learning things visually. I like learning things through sound. And they can be stable. So you might give me the same answer next week. But the trouble is, there doesn't seem to be any educational value in me knowing that as a teacher. because it does not mean that if I teach you in your preferred learning style, you will learn more quickly or better or more efficiently. And in fact, the neuroscience would not support that either. Because even if I say to you, if I showed you a picture of a bell, your auditory cortex and your temporal lobe will activate. Because you've seen a picture of a bell and you're imagining how a bell sounds, that's how interconnected the brain is. So this idea that simply by using a questionnaire, we can find out how you might learn more efficiently. There's no educational evidence for it. There's no neuroscience basis for it. And the few psychological studies that have been done have even found that there can be a benefit in you receiving information that is not in your preferred learning style. And there's new learning styles coming out all the time, and I'm very sorry to say there's no evidence to show they're of any educational value at all. Yes?
In the case of learning disabilities, though, is it not the case that, okay, it's hard to add, but it's easy to take away, so to speak? So in the case of barriers to learning, given that, for instance, sensory issues can be very different when you come to learning disabilities, distraction can be very easy. In this case, I think maybe that knowledge is useful.
I mean, I think if there is a damage to one modality, for example, I do believe that some individuals find better ways, find their own ways to learn and preferred ways of learning. So I'm not saying that when you've worked with a child, you find out that they're very good at learning in a particular way. I think that's what teachers do all the time.
But they can also be very distracted in another way. I think when this crosses over with learning styles and, of course, neuroscience.
I couldn't comment on that really, but all I would say is that I'm not saying that teachers should not be sensitive to the ways in which their pupils like to learn. And I'm certainly not saying that multimodality is a bad thing, because actually one good thing about learning styles is that it's encouraged teachers to try teaching things in an auditory, visual and kinesthetic manner. But we all benefit from receiving all modes, that's the thing. And that's why, that's why I passed the brain round, because I wanted you to see it, I wanted you to touch it, as well as just hear about it. I'll take one more question and then home for tea. Yes.
Wondering about what you said about you're lucky to be big primates.
Yes.
What then, evolutionary speaking, is the difference between us and sort of a big gorilla, or how did our brains or the early bipeds and then bring it to them?
Well, I think that comes down to teaching, actually. Education. Actually is probably the thing that is the thing that makes us most different to other species. It's the cultural transmission of knowledge. And in biological terms, the thing which is most remarkable about us, which actually forms the basis of education, is our interest in holding the attention of others. And this appears to be something which is uniquely human. So primates, especially the great apes, are particularly interested in other primates and great apes. And that's a primate thing, because primates are social, so they are interested in each other in a big way, right? But only humans are interested in others being interested in them. And when you have that's very important for teaching, because when I'm teaching you, I'm looking at you and I'm thinking, are you listening to me? And other primates don't do that. And we're beginning to we see this at a very early age, when a mother holds their little baby and they go, and that is totally, a human thing, and it's the beginning of a very important way of a very important, more sophisticated form of communication. And we're beginning to understand that that's what basically provided the springboard for more sophisticated cultural transmission of knowledge, which in its most formal sense is education, and therefore it's actually, education is the formal version of the thing that makes us most different to all other species. Possibly the only thing. Okay, well, on that rather epic note, we'll just say thank you very much for coming along. Look forward to seeing you next week. This is on YouTube very shortly. I hope you've enjoyed it.